So welcome back to the third and final Ulam lecture given by Nina Fedorov. What I want to do to start this third lecture off is to relate the first two of Nina's lectures to the work that you normally associate with the Santa Fe Institute, and that in particular the study of complex adaptive systems. So let me define the terms. So I think we all have an intuitive notion of what adaptation is. So that's a directed change in a system in response to changing environmental stimuli, where this change typically improves some objective function. Critical components of adaptation are learning and memory, such that individuals can improve their future performance based on their past history. That's adaptation. We also have an intuitive notion of complexity, and we define it typically by exclusion. You can think of a system on the one hand of being completely disordered and chaotic, and at the other hand highly ordered and almost crystalline, and somehow in the middle between those two poles of the spectrum is where we observe complexity. So at the Santa Fe Institute we spend a lot of time trying to invent quantitative methods using mathematics or using computer models to measure complexity in natural systems, and also develop simple dynamical models that manifest complexity based on the operation of very simple rules, such as the rules of development described by Nina. But of course, we don't restrict ourselves only to development, but also to economics and ecosystems and so forth. Now, one of the things that you become aware of when you work in these complex adaptive systems is there's always a risk of gravitating towards one of the two poles of that spectrum. On the one hand, if a system changes too slowly, becomes frozen, it becomes excessively regular and is unable to manifest interesting behavior. On the other hand, if the system changes too quickly, the entropy increases and it falls into a kind of chaotic disorder. The first of Nina's talks was really about adaptation, in the various responses to the environment. And the second talk was about models and mechanisms of genetic change that facilitate adaptation, but don't push change too fast that the system is incapable of repairing itself. And in fact, a number of questions from the audience yesterday related to that. How is it that the system itself can deal with such an elevated rate of change? I think the surprising thing about modern genetics is that biological systems sit much closer to the disordered side of that end of that spectrum than we thought was possible. So that brings us to tonight's lecture on genetically modified plants. And I suspect, actually thinking about this, and it's just a conjecture, that a lot of our unconscious fears relating to genetic modification have to do with a notion that we have that perhaps by accelerating the rate of natural plant evolution will cause the system to fall into that gravitational end of the spectrum associated with maximum disorder and become irreparably chaotic. Now, whereas the first two lectures were completely free of anthropic influences, the third one puts human beings squarely at the center. And, as I said, a species that often is rather myopic, and capable of rapidly accelerating natural rates of evolutionary change. The question is, how far are we free to modify nature? The only way to know this is through careful scientific experiment, free from the fetters of political and economic, not scrutiny, but interference, and with an eye firmly on the highest ethical standards. These are essentially the topics of today's lectures. Now, given the topic of the lecture, I'm sure that we're going to be quite divided as a community, and Nina represents one very well-informed position. Many of us, I should say, at the Institute don't necessarily share Nina's position. In fact, many of us have had some very heated debates where Nina typically comes out on top, I have to say, since she's typically much better informed than we are. But I would just like to, you might have seen some of the security here, I'd just like to say that if you do have a particularly strong opinion on the matter, wait until the discussion at the end of the lecture, because as you see, we have the table set up. We're going to give ourselves more time tonight to have a more lengthy discussion and be considerate of other people's opinions and other people's rights to ask questions. Finally, I'd like to thank Ginger Richardson for putting together the entire Ulam series. I don't know where are you, Ginger? Over there. So thanks again. I think we should give her a round of applause for doing such a good job. So, okay, with no more delays, you know.
What an introduction. That's a hard act to follow. I hope it's not a huge disappointment. So, to all of you who came before, welcome back. To all of you who are here the first time, welcome. Tonight we take on a very contemporary and a very contentious issue. Are GMOs the answer to the world's ongoing problems? Our numbers are expected to climb from slightly over 6 billion to a staggering 9 billion people in coming decades. Will GM foods create the doubly green revolution that the Rockefeller Foundation's Gordon Conway says will be needed to feed the world's people? Or are GMOs really a public menace, as many groups like Greenpeace would like you to think? Are they so new and so different and so unnatural that we have to fear the consequences of bringing them into our food supply? Is there evidence that they are harmful to us, to the environment? Okay, about me. I'm going to be perfectly upfront, so you know where I'm coming from. I'm a scientist. I'm a plant scientist, a geneticist, and a molecular biologist. I've had the extraordinary fortune to be a participant in developing the field of plant molecular genetics. I've watched with excitement and wonder as our ability to improve plants using molecular techniques has developed over recent decades. I've participated in the government's oversight of the applications of this technology. I served on the NIH Recombinant DNA Advisory Committee in the 1980s, as well as with groups convened by the Environmental Protection Agency, the EPA, the U.S. Department of Agriculture, USDA, and the Food and Drug Administration, the FDA, and the National Academy of Sciences. I've watched the growing worldwide controversy about GMOs with growing dismay. Several years ago, I decided if I were going to be the Lorax and speak for the trees, I'd better know what I was talking about. I learned a lot about the history of genetic modification of crops. I learned as much as I could about the concerns that people were raising. And then with the able assistance of a marvelous science writer, Nancy Brown, I wrote Mendel in the Kitchen. I can't tell you that everything in there is absolutely correct, but we sure tried our best to check and recheck our facts. I have just an hour with you, and I'll try to share the most important things I've learned, but the most important thing that I want to say to you about this, I can say right now. I would beg you to keep an open mind, keep asking questions, and keep seeking reliable information. Enough said. Let's start at the beginning. What is a GMO? What GMO means is genetically modified organism. Now today, that is almost exclusively used to designate an organism that was modified by molecular techniques. Now, is genetic modification new? The answer is no, it's not. People have been modifying plants to be better sources of food for themselves for somewhere between 15,000 and maybe even as much as 50,000 years. How do we know that? Well, let me give you some simple common sense examples. Wild plants have mechanisms to disperse their seeds. Think about the lovely parachute that a dandelion seed has to take it to a new place. Think about burrs. Those are simply seeds with hooks to hang on to animals, to disperse them. To be collected by people for food, the seeds have to stay on the plant. So a very early kind of mutation that people selected was one that prevents the plant from scattering its seeds. Now let me show you what that means. If I can get this to move. There we go. This is a wild grass called teosinte. It turns out to be what people turned into our modern corn plant, with its huge ear to which the kernel sticks so tightly that they can be collected in a whole nourishing package by a machine, a harvester. These two look so different than when people first discovered teosinte. They thought it was a different species. But it turns out that they are so closely related that they interbreed and they produce offspring that are everything in between from what the teosinte plant produces to the ear of corn. So this over here is the seed structure of the teosinte plant. And it has two characteristics that are absolutely, utterly useless for people. It has what is technically called a disarticulating rachis. That is, the seeds scatter when that structure dries down. And second, the seeds are hard as rock. Indeed, they have rock in them. They have silica deposits. And the reason for that is very simple. These creatures, the plants, use animals to disperse their seeds. And it doesn't do any good for the plant. It isn't able to disperse its seed if it's digested by the animal. So the point is that the seed has to survive through the digestive tract. And so the first thing that people select for is the ability to stick to the plant. Now, these structures that the teosinte plant makes are actually produced, technical challenge. There it is. At the top of the plant. This is really a grass plant. This thing is a shoot that normally grows out. It's actually telescoped. And if you think about an ear of corn with all the leaves on top of it, all of this is a telescoped shoot of the original teosinte plant. And so that's number one. And number two, the way that archaeologists can tell is that the mutations that cause the seeds to stick, that create, in technical jargon, the non-disarticulating rachis, are detectable even in archaeological samples. And even the earliest little tiny ears of corn that have been picked up from that date back 6,000 years already had pretty much this structure. So people selected for this very early on. And the other thing that's incredibly striking is that even though they didn't have trains and planes and trucks and all the mechanisms that we have for dispersing things, once people developed this plant, it spread in ancient terms extremely rapidly. So the genetic constitution of some of the strains that are in North America originated in Mexico, and they were already the same exact mutations are detectable in archaeological specimens from 3,000 years ago up in New Mexico, up in this area. So, going on, let's look at some other examples of genetic modifications. Here are some familiar foods that people think are quite natural, but this is a very bizarre mutation, actually. This is a head of cauliflower, of course, but what is this stuff? This stuff is a mutation that causes the plant to make flower buds and flower buds and flower buds and flower buds. It's just a stuttering mutant that can't get out of the loop of making flower buds. And then there's giant garlic. That was a gift of Luther Burbank. Luther Burbank actually was a remarkable person. He was one of the first plant breeders that understood the plasticity of plants and how much you can do by simply selecting nature's constant, from amongst nature's constant variety. And then there are tomatoes. Actually, this is from a very poisonous family, the nightshade family, and the wild relatives of tomatoes are rarely even red, And they certainly aren't these huge balloons that we think are tomatoes today. And then there's seedless oranges. Have you ever thought about a seedless orange? Seeds are the things that plants use to reproduce. We like our seedless oranges. All of the seedless oranges are propagated by people, by cloning. And then, of course, there's seedless grapefruits as well. But I want to tell you about how this particular strain, the Rio Red grapefruit, was developed. Lest you think genetic modification is new. So back in the last century, people developed ways of accelerating the rate of genetic change that is inherent in natural populations by virtue of cosmic rays and transposons. That is, part of the change is inherent in the organism, as all of you that came last night realize, and part of it is simply from cosmic radiation. People learn how to use chemical mutagens and radiation. And how this was created was by actually cutting a bunch of shoots of already slightly red, pinkish grapefruit, sending them off to Brookhaven National Laboratory where they were irradiated, and then shipped back to Texas and then grown for a number of years and looked at constantly. The breeders were looking for mutations, and they finally identified this as a mutant branch, propagated it, and released it some years later. Now, what I need to tell you is that that's not a very tidy process. Irradiation makes all kinds of mutations, and probably half or more of the varieties that are used today as plant foods have some irradiation mutagenesis in their background. Why does this work? Why didn't we regulate it? Largely because, and this is a little bit of an answer to David's challenge, largely because most of the mutations that happen are what the technical term is deleterious. They're harmful to the organism. But breeders learned that they could simply kill the plants that didn't look right. So it's a constant process of selecting the things that have the qualities and the growth properties that the breeders wanted. As I said earlier, the term GMO is used just for molecular modifications today. These are also called recombinant DNA techniques and genetic engineering, which seems like a very off-putting term. We don't have a problem with engineering cars or bridges or computers, but food? Isn't that fooling with Mother Nature? Well, as you just saw, over a few tens of thousands of years, we've done a pretty good job of cajoling mutants from Mother Nature to provide a vast array of plants that produce appealing and nourishing people foods. During the 20th century, we learned how to accelerate that process with chemicals and radiation to hurry along Mother Nature's slow pace of making mutations with cosmic rays and transposons and such. So what about the new molecular techniques? Well, here too we've learned some of Mother Nature's tricks and put them to work. Here's a thumbnail sketch of recombinant DNA technology. Bacteria have, in addition to their big chromosomes, these little mini chromosomes called plasmids. Bacteria also make a large array of DNA cutting enzymes called restriction enzymes. The EcoR1 you see in this diagram is such a restriction enzyme. And it cuts DNA in the way that it leaves overhanging and sticky ends. Imagine two pieces of Velcro and you leave the ends hanging over. You can stick them together. And that's basically what's happening here. So the purple piece of DNA is from a different source, perhaps a corn plant or a fruit fly. The plasmid is cut open and the DNA is cut up with the same restriction enzyme and stuck together. And there's one more piece. There's an enzyme which is appropriately called a ligase that ligates this thing together. And then those plasmids are introduced into bacteria. The bacteria are first treated with a little calcium chloride, which makes them take up the DNA. for reasons nobody really understands. And then those bacteria with the plasmids in them are spread out. Now you see 3 plasmids here, but in fact, it's done in such a way that most cells have just 1 plasmid. And then those cells are spread out on a Petri dish, like this, that selects those particular plasmids that received the bacteria that received the plasmids, and it looks like this in reality. And each one of those single cells containing a single plasmid with a unique sequence grows into many millions of cells that constitute the little colony. Each one is a different cloned piece of DNA. Basically, it's like making many, many copies of some kind of a document. And that gives biochemists, or molecular biologists as they're called now, large numbers of copies of it. And it also produces the protein it codes for. But this basically allows the biochemist to analyze both the structure of the DNA and to modify it. Now, today, for example, people are treated with human insulin that's produced in just this way. A human insulin gene was inserted into a plasmid, which then makes very large numbers of copies of it, and in turn produces the protein it codes for. This has been a tremendous medical advance. Before recombinant insulin, people were usually treated with pig insulin. With diabetes a growing problem worldwide, it would have been very difficult to keep up with the demand. And because it's a foreign protein, people often reacted badly to pig insulin. That's something we accept today, but I will tell you that back in the 70s, there was a good deal of furor about it, even that well-established technology. Now, to understand contemporary molecular approaches to modifying crop plants, you need to meet one more actor, nature's own genetic engineer, a soil bacterium called Agrobacterium tumefaciens. This bacterium causes the formation of galls, which are tumors, on the stems and roots of many plants. The understanding of how the bacterium forms the galls, the pioneering work that made it possible to use Agrobacterium to carry new genes into plants was done by the late Jeff Shell, a dear friend and a fantastic scientist. In fact, I think this picture came from him back when we were collaborating. The animation that I will show you next gives you an idea of what was understood through basic research about the gall formation in a root. This clever creature carries around plant genes that it doesn't use. But when it detects an opening, meaning a wounded plant cell, it swims over to it and slips those genes into the plant. These genes reprogram the plant a bit. First, to grow this tumor, and second, to make a special compound that the bacteria in the vicinity can use for nourishment. Very clever. The tumor doesn't kill the plant, it just coaxes it to make food for the bacterium. So this is a little animation. The source of the animation, by the way, is immediately under it. That represents the chemical signals sent out by the wound, and then the bacterium detects those and swims up the concentration gradient. It has these lovely little flagella that let it swim along. And then when it gets to the actual cell, it attaches, and it's carrying a plasmid of its own, and then it transfers a piece of that plasmid into the cell. Here it comes again. And then a most remarkable thing happens. There's a mechanism that actually cuts the DNA and allows the DNA to integrate. And that, as I said, reprograms the cell to make this little tumor, which then produces compounds called opines, nopalene, octapine. There's a bunch of different ones, but they can serve as a sole food source for the bacterium in the vicinity. So here's the little gall. It doesn't kill the plant, it just feeds the bacterium. Kind of an interesting process. So what Shell and others did was basically to remove the cancer-forming genes, the gall-forming genes. The gall is the name for the tumor. And then they developed ways of incorporating a new gene, putting Agrobacterium to work as a gene delivery system in order to improve crop plants. Here's what it looks like in the laboratory. So you take a leaf. This is actually a tobacco leaf, not because it's tobacco, but it turns out that tobacco is particularly easy to transform, and so many laboratories use it as a test system. So you cut up the leaves, and you dip them in a bacterial culture. That cutting them up causes them to release the chemicals that stimulate the bacterium to transfer its DNA. The leaf pieces are simply dipped in a bacterial suspension, then a couple of days later, they're transferred to a medium that contains an antibiotic to eliminate the bacteria, and a compound that permits only the cells that receive the DNA to grow. The medium also contains plant hormones that stimulate the cells to grow in these little lumps. These are called calli. You can see these little white lumps. Now, the miracle of plants is that if you take those hormones away, that lumpy thing begins to make leaves. And then it begins to look like a plant. And then it makes roots. It develops into a perfectly normal looking plant, but now it has the gene that you added. Quite remarkable. Okay, that's the basic biology of it. Now let's look at some of the things that have been done with these molecular techniques to protect crop plants from diseases and insects. This plant is infected with papaya ring spot virus. You could easily see how the virus got its name by the spots. Now, that papaya doesn't look bad, but here's what it does to plants. This is a healthy plant, and this is a virus-infected plant. Here's the critter that carries it, or here's the virus itself. It's basically a piece of nucleic acid, in this case RNA, for those of you who came last night, in a protein coat. And here's the little insect that carries it around. When it sucks fluids from the leaf, this is actually leaf that it's sitting on, and it sucks up virus particles and spreads it to the next plant that it lands on. Now, the virus, abbreviated PRSV, was first discovered in Hawaii back in the 1940s. By the 1950s, it had basically wiped out the Oahu's papaya industry, which then moved to the Big Island and was kept relatively free of the virus for several decades by constant vigilance and rapid elimination of affected trees. This is a very difficult virus to control because you simply can't kill every insect that's flying through an orchard. It's extremely difficult. And people had spent years and many millions of dollars trying to develop resistant varieties with very little success. Now, the unlikely hero of this story is a plant pathologist by the name of Dennis Gonsalves. He had worked for many, many years. He was very familiar with the disease. He'd worked on it for many years, as I said, with very little success. But back in the late 70s, he heard that a colleague in St. Louis, Roger Beachy, had used the kinds of techniques I just described to you to introduce a viral gene into tomatoes and found that it protected them from infection. Now, I'll show you how this actually works, but the original idea was that maybe, like animals, you could inoculate a plant against a disease. And that wasn't such a far-fetched idea, actually, because it rested on observations going very far back by plant pathologists, that once a field or a plant had been infected with one virus, it was now cross-protected against superinfection with another virus. That's called cross-protection. So now he decided, this is Gonsalves, tried to try out this new molecular approach. Now, papaya, unfortunately, isn't as easy to transform with agrobacterium as many other plants are. But he persuaded a colleague of his at Cornell, John Sanford, to try using his newly invented gadget that he called a gene gun to introduce viral genes into papaya plants. This so-called gene gun actually shoots tiny, tiny particles of gold coated with DNA through the plant cell wall. Plant cells are enclosed in a box, in fact. And that makes them very difficult to introduce DNA to. But it worked. And when the little particles deposit their cargo, some of it goes into the nucleus and gets incorporated into DNA. Now, it's not such an easy technique to do, and it took them more than a decade. But by 1991, the first virus-resistant plants were ready. And by 1992, they'd received permission to carry out the first field trials in Hawaii in collaboration with a group at the University of Hawaii. Now, just about this time, the virus was beginning to get really out of control on the Big Island. But by 1994, the USDA had approved the first large-scale field tests. And in fact, the USDA wisely gave them permission to actually grow seeds because they knew that it would take quite a long time to get through the whole regulatory apparatus, which included safety testing required by the USDA, the EPA, and the FDA. And that actually took 3 years. And by that time, the virus was bad enough that farmers were going out of business. Finally, the approvals were obtained, all the safety testing was met, and the seeds were finally released in 1998. Free of charge to growers. This was an academic project. There aren't many of them because there aren't many groups that can afford to keep going through all of this. But by about 2000, the Hawaiian industry had started to bounce back, and the production levels had gone back up to pre-1995 levels. Now here are the healthy trees that carry the viral gene. To me, this is a remarkable story. It's as remarkable as the history of vaccination and medicine. And indeed, carrying the viral gene, in essence, vaccinates the papaya plant against the virus, only by a very different mechanism, which has only been understood in the last few years. When they actually did this work, they knew it worked, but they didn't know why. Let me show you how it works. A virus, as I said earlier, is simply a packet of genes, in this case born on an RNA molecule, in a protein coat. I've represented the protein coat in these green ovals. And the first thing that happens when the virus is delivered into the cell is that it gets undressed. And that allows the nucleic acid, the RNA, to be replicated. And that RNA then codes for proteins. Those are more coat proteins. And that allows the virus to reassemble. And it's pretty much ready to be picked up by the next insect that comes along. But meanwhile, it's pretty much trashed the place. And eventually, such infection will kill the plant, as I showed you. Now, what happens in these plants that have a viral gene is quite remarkably different. So the minute that the protein coat is removed, there's a mechanism that triggers the destruction of the RNA, so it never gets off the ground. Well, is this something terrible that never happens in nature? No. It's exactly how plants become immune to viral infection. In fact, very similar, at least at a formal level, to how you become immune to disease once you've had it. The next time the same virus is seen by your immune system, it recognizes it and destroys it. And so it is with plants. Once they've been infected with a virus, this mechanism allows them to resist reinfection. What Gonzalez and his colleagues did then was figure out how to make the papaya tree immune to the ring spot virus by expressing just a little piece of the viral genetic information, enough to trigger the mechanism, but not enough to make the plant sick. Thinks it's been infected, even though it hasn't been. He's considered a hero by the local farmers and has received national and international awards for this work. But he's also been attacked and vilified for it by people and groups that are against it. On to corn and insect resistance. Here's a healthy ear of corn, and here's a very common corn pest, the European corn borer, and what it does to the corn. Its most devastating effect is to hollow out the stalk so that the plant falls over and dies. Several biotechnology companies, the most familiar of which to you probably would be Monsanto, transferred a gene from a soil bacterium called Bacillus thuringiensis, abbreviated BT, which is why it's called BT corn, to plants. The bacterium itself has been used for insect control for many decades because it produces crystals of a protein that is toxic to certain kind of insects, but perfectly innocuous to people. It's just another bit of protein to people. It's been sprayed from helicopters to control gypsy moths. Organic farmers use it extensively. You can get it at the hardware store under a number of brand labels, including Dipel. In fact, it's considered so safe that the EPA hasn't set what it called tolerances for it. The EPA's tolerance is the maximum level of chemical, such as a pesticide or fungicide, that is considered safe for human health as a residue in food. BT corn is resistant to the corn borer because the plant itself expresses that protein. This means that farmers don't have to use chemical pesticides to control this pest, which is extremely difficult to control because of its boring lifestyle. The rapid adoption of BT corn and cotton by farmers in many countries has actually dramatically reduced pesticide use. But there turns out to be an unexpected additional benefit of BT corn, which people really didn't anticipate. Invading corn borers provide a path of infection for fungi, the most serious of which are Fusarium ear rot and kernel rot. These fungi, which make the white stuff you can see on the upper ear, also produce some extremely toxic compounds called fumonisins and aflatoxin, which cause serious neurological diseases and death in animals and people. The use of BT corn dramatically reduces the incidence of such contamination by such fungal toxins. A gift, unexpected. Let's talk a bit about golden rice, rice that is yellow because it makes more vitamin A. Now here's this tale, or Ingo Petrikas, a Swiss plant biologist that you saw on an early slide, and a biochemist, Peter Beyer, who collaborated to develop vitamin A-rich rice. Ingo took up the project more than 15 years ago because he himself had experienced hunger as a child, having lost his father in the Second World War, and he wanted to address a real problem that affected real people who were much less fortunate than he. At a conference called together by the Rockefeller Foundation, which funds a great deal of agricultural research, the attendees agreed that adding the ability to produce vitamin A to rice, a major part of the diet of many people in poorer countries, would have huge health benefits. Many children die of vitamin A deficiency, and adults have too little of it, can go blind. Now, Ingo was determined that whatever he produced would be freely available to farmers everywhere. Being a somewhat naive academic scientist, he was rather chagrined to discover that he'd violated some 70 patents held by companies, and the rice couldn't be made available immediately. But eventually that problem was also solved, and most of the companies actually donated their patent licenses to this project. Today, that project is overseen by a group called the Golden Rice Humanitarian Board, which is chaired by the now retired Professor Petrikas. Ingo also admits to being shocked at the extent to which he was vilified by groups and individuals who are against using molecular techniques. Now, currently, the rice is being improved and field tested. It, too, must pass through a long regulatory process before it is released, in addition to the field tests for how well it stands up under various growing conditions. It's also being tested for the bioavailability of vitamin A precursor and simply for palatability, how good it tastes. Interestingly, a recent breakthrough at Syngenta will allow a much higher level of beta-carotene expression than was previously possible. One of the things that Inga was quite viciously attacked about was that, gee, people would have to eat, 40 pounds of rice a day in order to get enough vitamin A, and that this was a sham. But in fact, those problems are being solved. And eventually, these particular genes will be, then they have to be transferred to rice varieties that are particularly well adapted to various parts of the world. I suspect that this will eventually be released. The question will be, will people accept it? We don't always make decisions about foods based on what's best for us. And people really value the whiteness of rice. So that's the next challenge. Let's go on now to consider some of the concerns people express. Is it safe to eat DNA? Now, this is a very sophisticated audience, but I have to tell you that in surveys, a surprising number of people think that the only foods that contain DNA are those that have been genetically modified by these techniques. Well, it turns out that all food contains DNA. The amount of extra DNA introduced by molecular methods is about 1 part in a million, or 0.0001%. DNA digestion begins in the mouth and is completed in the stomach and the digestive tract, although small bits can survive. This doesn't change our genes. You've been eating a lot of foreign DNA all of your life, both plant and animal DNA. The next question is this: could GM foods contain new toxins? Well, all foods contain chemicals that are toxic or carcinogenic, or both, but generally only if you concerned large quantities. Now, crop plants are plants. You all know that there are poisonous plants, and crop plants are plants which people have over millennia either bred for decreased toxicity, or in some case figured out how to reduce the toxicity by cooking or some other treatment. Nonetheless, plant breeding, both traditional and contemporary, can change the abundance of a toxic compound. Basically, animals protect themselves from insects and bacteria through their chemical arsenals. Now, this has happened by traditional breeding. There are 2 very well-known cases where new conventionally bred insect-resistant varieties of potato and celery turned out to have elevated concentrations of compounds that were really toxic to human beings. But that was before any kind of testing was done, and those particular varieties were pulled off the market after they were released. As I said, plants protect themselves from insects, animals, and pathogens by a variety of strategies, some of which include producing quite toxic chemicals. But the good news is that the ability to analyze the complete chemical composition of a crop has gotten extremely good, and before a crop plant is introduced, the developer submits information that compares the chemical composition of the original variety with that of the modified variety. Okay, but that only applies to those varieties that were modified by molecular techniques. Curious. Next question is this: could GM foods contain new allergens? Now, most allergies are caused by a handful of proteins, and the amount of information available about allergens is increasing all the time, but we don't understand it completely. Each protein for which genetic instructions are being introduced must be tested at pretty high concentrations, both for toxicity and allergenicity. And if there's any slight indication that it could be an allergen, or even if the gene comes from a plant, like a peanut plant, which does produce a common allergen, it must be tested for allergenicity. So what I can tell you is that GM foods are tested more than any crop ever produced by conventional breeding, chemical mutagenesis, or radiation mutagenesis was ever tested. By the time it's passed through all the tests, a GM food is frankly no more likely to cause a problem than its non-GM progenitor. Another thing people worry about is genes escaping and causing problems. Gene transfer from crops to other plants is now called gene flow. What you need to know, and it's just common sense, is that plants have to belong to the same species in order to trade genes. Plant genes travel two ways, either in seeds or in pollen, which is carried by wind and by insects. Cross-pollination, which is what it used to be called, is frankly a familiar problem to farmers, and particularly for seed producers. For example, if you grow a little patch of sweet corn in the middle of your fodder corn, you're not going to get sweet corn. You'll get starchy fodder corn. Sweet corn's a mutant. For those of you who came last night, it's a recessive mutant that can't convert the sugar it collects in the kernels to the normal storage form, which is starch. Pollen made by the fodder corn will contain the normal genes that code for the enzymes that convert sugar to starch. So you have to keep them quite separate. Another example is rapeseed and canola. Canola oil is regarded as one of the most healthful oils today. Canola is a simple mutant derivative of rapeseed. Rapeseed oil is high in something called erucic acid, which turned out not to be very good for people. Canola is simply rapeseed that has a mutation that prevents the production of the erucic acid. Rapeseed and canola are very closely related plants, and farmers growing both have to be keenly mindful of keeping them separate. So this is not a new problem for farmers. Whether genes can escape into wild plants depends on the plant. Whether it matters depends on the trait. Corn, for example, has wild relatives in Central and South America, but not in North America, or most places that it's grown around the world. So the first requirement is that there be a local weed species that can cross-hybridize with the plant in question. Then the other issue is the trait itself. Herbicide resistance, for example, is a potential management problem for farmers, but probably not a big concern in the wild where herbicides are not used. So the answer to the first question is, it simply depends. There's not a blanket answer. You have to look at the crop, the adjacent, what grows in the immediate vicinity, and what the trait is. These are the kinds of issues that the EPA and the USDA worry about when they examine new crops for release. Now, will GM crops reduce biodiversity? The biodiversity issue has different sides to it. Some people mean crop diversity, and others mean the diversity of species other than the crop plants, and both are significant issues, but rather different. Crop biodiversity issue was much in the news in recent years when Bt genes, which came from a bacterium, were introduced into corn by molecular techniques by companies like Monsanto, as I described to you earlier. And they were discovered in Mexican land races. These are the beautiful, colorful strains that are a specialty in many areas of Mexico. But the fact of the matter is that farmers grow these beautiful strains, are constantly selecting them for how they look and how they grow. If they were able to derive an insect-resistant line, it would actually help them and their ability to maintain these strains. Their biggest problem would be the patents surrounding the Bt gene. Now, another story that captured media headlines a few years back was that the larvae of monarch butterflies died when fed Bt corn pollen in the laboratory. It's not a big surprise, since these butterflies are closely related to insects that the Bt was introduced to control. But butterflies actually don't eat pollen. They eat milkweed, which generally doesn't grow in farmers' fields. Nonetheless, in response to the worldwide concern that this story aroused, a very large study was done to assess the potential impact of Bt pollen on monarch butterfly larvae in the field. The study stretched from Maryland to Canada and measured everything imaginable. How far the corn pollen goes from the fields, how much milkweed is in them, where it falls on the milkweed plant, so forth. The results were published in 5 back-to-back papers in the proceedings of the National Academy of Sciences. Their conclusion was, in the worst-case scenario they could construct, only about 1 of 2,500 larvae might be affected. That compares with about 90% or more death rate caused by conventional pesticides. Needless to say, this did not make headlines around the world. On the larger issue of biodiversity, that is the biodiversity that's preserved in wild ecosystems of all kinds, I like to quote the words of Peter Raven. He's the director of the Missouri Botanical Garden and a tireless champion of biodiversity around the world. He says, nothing has driven more species to extinction or caused more instability in the world's ecosystems than the development of an agriculture sufficient to feed 6.3 billion people. It's important to know the following to get your arms around what this means for biodiversity. First, the world's human population, as I said much earlier, is expected to grow somewhere in the neighborhood of 9 billion people in the coming decades before it stops growing. Second, many people know that fact, but second, the amount of farmland actually in cultivation hasn't changed in 50 years. We are losing good cropland to desertification, salinization, and urbanization as fast as we're adding it by chopping down species-rich tropical forests. And asphalt, as someone said so eloquently, is the land's last crop. The choice comes down to biodiversity or food, unless we can do more with less. Raven says, to assert that GM techniques are a threat to biodiversity is to state the exact opposite of the truth. The less focused and productive this agriculture is, the more destructive its effects will be. What he's saying is simply that if we want to preserve biodiversity, we have to get better at growing food on the land that we already cultivate, and we have to get quite a lot better at it. Well, okay. If all that I've been saying is true, why is there so much resistance to the new molecular methods of crop modification? Here, I'm not sure I have a great answer, but what I can do is show you an example from the past, and it's not a unique example. Today, we consider the elimination of the scourge of smallpox through vaccination as a real achievement of mankind. This is a disease that kills a third of those it infects and disfigures virtually all the rest. But back when governments began to compel their citizens to be vaccinated, resistance to the procedure was considerable. Anti-vaccination societies, which by the way persist today, became especially vocal during the late 19th century. Many anti-vaccinators believed that vaccination was, as George Bernard Shaw put it, a filthy piece of witchcraft, which did more harm than good. Opponents of compulsory vaccination were varied. Some saw the issue in terms of civil liberties, arguing that governments should not force citizens to undergo any medical treatment against their will. Others believed vaccination was dangerous, insisting that thousands are killed annually by vaccination. And still others, especially in India, found the use of vaccines derived from cows to be unacceptable. The story of how the disease was finally eradicated through persuasion, and even sometimes by a bit of force, is a fascinating one. It has actually many parallels to the present controversy about GMO foods. Here's a cartoon from the early 1800s. It shows people turning into cows because of the vaccination. That's because of cowpox virus. Milkmaids who were exposed to the disease, this particular disease of cows, didn't get smallpox. People eventually figured out that cowpox virus cross-protected people from smallpox virus without making them sick. So they started inoculating people with cowpox virus, hence the cartoon. People were worried that the cowpox virus would turn them into cows. So why does it matter whether we accept and use these methods? Why can't we just do what we're doing? Well, let me share the words of the Kenyan Florence Wambugu with you. Let me tell you a little bit about her first. When she was 13, even though the food was rather scarce, her mother sold the family cow to pay for Florence to go to a boarding school 10 miles away. Her mother needed permission from the village council to sell the cow, and most people thought she was crazy for educating a daughter rather than marrying her off. Wambugu got a PhD in England in plant pathology and went back to Africa for a number of years to work on deriving a virus-resistant strain of sweet potato. Here's a healthy one. Here's a sick one. It's a staple part in that part of the world. Sweet potatoes can be counted on when much else fails. Then she went to do research in the US and was actually invited to Monsanto. At Monsanto, she used the same kinds of methods that I described to you for papayas to develop a virus-resistant potato variety, which she took back to Africa with Monsanto's blessing. They donated the technology to Africa. Today, Wambugu lives and works in Nairobi, championing the importance of biotechnology to Africa. The virus-resistant sweet potatoes are being grown on the outskirts of Nairobi, according to a New York Times article last month, but haven't been released yet to farmers. Wambugu faces some difficult obstacles. Groups from Greenpeace to the Union of Concerned Scientists fret that Africa is being manipulated by multinational corporations, in effect serving as big and potentially dangerous genetic experiments. Some Africans are so worried about plant biotechnology, for example, Zambia's government would rather have its citizens starve than accept donated food that includes genetically modified corn. Concerned with these misunderstandings about transgenic plants, Wambugu created the Harvest Biotech Foundation International to serve as a Pan-African voice on the issue. Wambugu firmly believes that the potential good far outweighs the risk. Like vaccination or penicillin, there are a few people who are allergic or have a bad reaction, but both do, on balance, far more good than harm. She says, you people in the developed world are free to discuss the merits of genetically modified foods. But can we eat first? Thank you for your time and attention. Oh, now come the hard questions.
No, yes, I prepared. Can you hear me? No? Is this on? Can you hear me now?
I don't think they've turned it on yet.
Maybe if I turn it on.
Yes, it might work better.
Okay, there you go. Right, I think we should heat things up just slightly. Beautiful talk, and very convincing, and a real education. But, I mean, I sometimes think, actually, the reason you become an academic is so that you can get paid to argue. And so let's do that. Is it gone off again?
No.
So the talk could be accused of being slightly one-sided.
Guilty.
Okay, so let's just introduce. So you mentioned the BT corn. So any one of us here can go to Medline or Google Scholar and do a search on recent studies on BT corn, which I did. And it's absolutely true that it's a complete mess. Half of the studies claim that there's no demonstrable deleterious effect. And another half say there very definitely is. And I could quote them, but the point here would be that presumably we all live by this dictum, right, that absence of evidence is not evidence of absence. Given that there is some ambiguity remaining as to the possible harmful effects of BT corn, is it advisable to release it into the wild, where once we've released it, we have limited control?
That actually has a lot of questions embedded in it. I can't comment on those studies because there are good studies and bad studies. And unfortunately, a lot of the studies, just as in the drug industry, because a lot of the development happens inside of companies, the studies aren't released. So I think, first of all, you have to look at the quality of the study. And second, the other part of your question is release into the wild. Crop plants don't live in the wild. Corn seeds, as I tried to show you, are attached so firmly that even if a whole ear stays in the field, the kernels germinate right there. You see, for example, when farmers rotate corn and soybeans, you'll see an occasional corn plant sticking up through them. Those are called volunteers, and they're a problem. But it's not the day of the triffids. They don't jump the fence and take over the world.
Well, we can talk about jumping the fence in a second. But there is this problem of, you talked a little bit about this issue that sometimes gets called non-target specificity or target specificity. And in a number of studies, there was the observation that, so you might provide a plant with a toxin that is intended to target Coleoptera, for example.
But also would kill Lepidoptera. I gave you that example.
Right, and also phylogenetically quite distant groups like Neurospora. And so one of the things I think that... Neurospora.
Oh, Neurospora is a fungus, my dear.
No, the group lacewings, the lacewings.
Oh.
Can't hear you.
Sorry.
Speak into this.
Yeah, it's a little bit tricky. I should perhaps
Speak up.
hold it here. I am actually speaking up, believe it or not. So, no.
It's just that his questions are so complicated.
Yeah, they are. I mean, there are more distantly related insect groups like the lacewings that also were sensitive to the genetically modified plants, whereas the original intention was to target exclusively, if possible, the Coleoptera. And so I think that late 90s, the discovery that the confidence people had in their ability to target specific insect groups was probably misplaced.
I don't think entomologists were particularly unaware of the target specificity of BT. But again, I think you have to put it in context. If you spray a pesticide, a chemical pesticide on the field, you kill somewhere between 90 and 95% of what lives in that field. If you put the toxin gene in the plant, it's only the insects that munch on the plant. So the death rate is much lower. I think that people are simply unaware of the use of pesticides. Anything that will reduce pesticide use really is beneficial. Even though, I mean, pesticides are very much less specific.
Well, no, but presumably if the GM plant were to become ubiquitous, then the deleterious effects would also be highly non-localized. So even though it's true that it's only the insects munching on the plant, if those are the only plants we possess, then all of the insects have no option but to munch on them.
It's not clear to me how you would accomplish that. I mean, the problem is that I think what you're missing is that crop plants are dependent on people for propagation. Okay, we have made sure that that's true over.
I can hear you, and I'm an organic farmer, and I have a question about the.
Well, perhaps if you wait, then I might ask you. Let me just.
Can't hear you. So basically.
Yeah. Okay. So something wrong with the mics.
You're mumbling.
No, you need to mumble.
We're not. We really are not mumbling. It's an English accent. Okay, let me just one question, Nina, because before we go to the questions, there's jumping the fence. So you mentioned the case of transgenically modified plants being found in Oaxaca. Now, Mexico bans transgenically modified plants, And so this is a highly non-local, what would be called a global propagation of a GM plant. The other case, which is quite well known, is the so-called Starlink gene. And the Starlink gene was created by the company called Aventis Crop Sciences. It's a French company, and this gene has infiltrated, introgressed, as we call it, into American plants. And over 5 years there have been very deliberative attempts to remove that gene from American plants with very little success. Now, given that Aventis owns the patent, there are all sorts of thorny issues related to who owns the plant that that particular gene has introgressed into. So how do we deal with a world where all of these large companies are claiming ownership based on the introgression of genes which local farmers had no ability to control?
Thank you. Yeah.
There are many issues that you've articulated there, and introgression, that's kind of an interesting, I can't take them all because they're all sort of in a big pot here. Can I take one piece at a time?
Yeah.
Okay. So the claim in the case of the Mexican varieties, the original publication claimed exactly what you said. Introgression is when a gene actually enters and becomes part of the genetic makeup such that all the other stuff is gone. That takes multiple generations of back crossing to get rid of all the other strains. That isn't the way crop plants work, okay? For the most part, farmers today buy their seeds, not because they have to, they can plant whatever they want to, but because they are guaranteed disease resistant, better, yield more, etc. That's, particularly in corn, hybrid corn isn't kept because the hybrid vigor, which comes from the whole breeding process, breaks down very quickly. Now, there's this whole stuff about introgression, but so far, that, let me go back to the first thing that you cited, which was the claim that there were genes introgressed into the local corn. Introgression takes many, many generations, and that simply, people took that data and reanalyzed it, and what they realized was that what the scientists had identified as introgression was just simply erroneous magnification of genes. They simply analyzed, they should have been able to figure that out. The paper was retracted, all kinds of stuff like that. Nonetheless, it's almost inevitable that that gene will enter the population. That's more of a problem for Monsanto than anything else, or for the companies, right? Than it is for the people. Because if it's a beneficial gene, especially that beautiful colored corn, if you still had all the traits and you introgressed a disease resistance gene or an insect resistant gene, you'd be in a better place.
If you owned it.
If you owned it, okay. And in fact, that's a problem for the company. Now, the companies generally, you know, it's expensive to sue. And the cases that have come up, there's a very famous one in a canola farmer who actually propagated. I think he was going to go into the business of doing it. His name is Percy Schmeiser. And a number of courts looked at all the evidence, and they decided that he was deliberately doing it. So they ruled against him. But Monsanto isn't going to go after little farmers that this happens to. It's just, they aren't going to get anything out of it. It's that simple.
All right, well, thank you for graciously taking thorny questions. I think
please,
I think, Nina, you were going to wander around.
Yeah.
Would you like to ask your question now?
Yeah.
Please do.
I also want to say Percy Schmeiser was perfecting his canola seed for 15 years. It became contaminated by Monsanto's, and they sued him for piracy. That's my understanding of the story from Percy Schmeiser's own writing. But I
May I please say that in fact it went through several court trials, and the seeds were tested. It turned out that, that he didn't know it, but he had actually taken his seed for testing, and sometimes the testing companies keep a little of that seed. And when that was reanalyzed, it turned out that a very large fraction of it was herbicide resistant. And the only way that you can do that is by spraying herbicide and picking the seeds that survive. Sorry, he did it.
Another question?
Yes.
No, from another person.
I asked about BT. That was my original question.
Well,
I'm an organic farmer. BT is a very useful tool to organic farmers. And when the BT is bred into the plants, bugs have very short lifespans that, they go through several generations in a growing season. Doesn't that make it less effective, the BT become less effective because there's more bugs that are generated that have that resistance to the effects of it?
That was a major concern originally. And so anticipating that that might happen, what companies designed was an agreement between farmers and companies that they would set aside what are called refugia, areas that are planted in non-BT crops, the principle being that if any insect-resistant survivor from the BT region would have a higher probability of breeding with an insect from the BT non-resistant population, and that would decrease the rate at which resistance arose. Now, people worried a lot about this, and it turned out that the compliance with those, some farmers didn't even know about it, the compliance of it was probably less than anybody expected, certainly in the early years. But a recent study completed within the last couple of years by entomologists showed that the BT resistance hadn't increased. And that was a real surprise. In fact, the entomologist who was the principal investigator on that said in a press conference that, if he had said that 7 years earlier, nobody would have believed him. So the bottom line is that it actually is not as big a problem as people thought originally, number one. Number two, even given that it turns out to be not as big a problem, what companies have started to do, which is a smart idea, is to double up on those resistance genes so that the insect would have to have 2 independent mutations to become resistant. Now, the probability of a mutation per generation and so forth is pretty small, like 1 in a million. But if you go 1 in a million times 1 in a million, that's a very, very small number. So actually, it is not the case that this will never happen. It is the case that, unlike pesticides, which of course do the same thing, you get pesticide resistance, people are thinking more about it in order to make this a longer cycle until you have to reinvent it. Next. Yes?
I had heard with genetically modified crops, there's not reproducibility. That, after the crop grows 1 year, then you have to go buy seeds the next year. Is that true or not?
The question is there reproducibility of genetically modified crops? Can you grow them? Actually, the answer to that, particularly with corn, is that since the early 30s, farmers pretty much have gone over to buying hybrid corn. Now, that is an invention that the science of which goes back to the turn of the century, when it was discovered that inbred varieties, which made puny little plants, if crossed, made big, healthy plants in the first generation. But that all falls apart subsequently. Now, interestingly, the history of the introduction of hybrid varieties was resisted by farmers. They said, gee, we don't want to be dependent on companies. We just want to keep our seed and grow it. Now, the head of the, a retired gentleman by the name of Don Duvik, who was the head of the plant breeding department of Pioneer Hybrid, sent me the following story by email when I was writing my book. He said, you know, my dad, like everybody else, resisted introducing hybrid corn, but back in 1936, there was a severe drought, and the only thing, he said, that was worth bringing in was the hybrid corn, and that converted farmers to planting hybrid corn, and then it took off.
Another question?
Yes.
No, from another person.
I asked about BT. That was my original question.
Well, is it?
I'm an organic farmer. BT is a very useful tool to organic farmers. And when the BT is bred into the plants, bugs have very short lifespans that, you know, they go through several generations in a growing season. Doesn't that make it less effective, the BT become less effective because there's more bugs that are generated that have that resistance to the effects of it?
That was a major concern originally. And so, anticipating that that might happen, what companies designed was an agreement between farmers and companies that they would set aside what are called refugia, areas that are planted in non-BT crops. The principle being that if the any insect-resistant survivor from the BT region would have a higher probability of breeding with an insect from the BT non-resistant population, and that would decrease the rate at which resistance arose. Now, people worried a lot about this, and it turned out that the compliance with those, some farmers didn't even know about it, the compliance of it was probably less than anybody expected, certainly in the early years. But a recent study completed within the last couple of years by entomologists showed that the BT resistance hadn't increased. And that was a real surprise. In fact, the entomologist who was the principal investigator on that said in a press conference that, you know, if he had said that 7 years earlier, nobody would have believed him. So the bottom line is that it actually is not as big a problem as people thought originally, number one. Number two, even given that it turns out to be not as big a problem, what companies have started to do, which is a smart idea, is to double up on those resistance genes so that the insect would have to have 2 independent mutations to become resistant. Now, the probability of a mutation per generation and so forth is pretty small, like 1 in a million. But if you go 1 in a million times 1 in a million, that's a very, very small number. So actually, it is not the case that this will never happen. It is the case that, unlike pesticides, which of course do the same thing, you get pesticide resistance, people are thinking more about it in order to make this a longer cycle until you have to reinvent it. Next. Yes.
I heard with genetically modified crops, there's not reproducibility. That, you know, after the crop grows 1 year, then you have to go buy seeds the next year. Is that true or not?
The question is there reproducibility of genetically modified crops? Can you grow them? Actually, the answer to that, particularly with corn, is that since the early 30s, farmers pretty much have gone over to buying hybrid corn. Now, that is an invention that the science of which goes back to the turn of the century, when it was discovered that inbred varieties, which made puny little plants, if crossed, made big, healthy plants in the first generation. But that all falls apart subsequently. Now, interestingly, the history of the introduction of hybrid varieties was resisted by farmers. They said, gee, we don't want to be dependent on companies. We just want to keep our seed and grow it. Now, the head of the a retired gentleman by the name of Don Duvik, who was the head of the plant breeding department of Pioneer Hybrid, sent me the following story by email when I was writing my book. He said, you know, my dad, like everybody else, resisted introducing hybrid corn, but back in 1936, there was a severe drought, and the only thing, he said, that was worth bringing in was the hybrid corn, and that converted farmers to planting hybrid corn, and then it took off. Today, almost all of it is hybrid. But then he had a little footnote to that. He said, you know, the next year, my dad kept some of that corn and he planted it out. And he said, my dad said, I just wanted to know if the professors were right. He said it wasn't worth even bringing in. So that hybrid vigor, which people really don't even understand yet, is what you see growing out when you travel across the country and you see these fantastically productive fields of corn.
But if you kept that corn kernels, could you plant them the next year?
Of course you can plant them. You would not have the yield that you had in the previous generation. That's the point. Yes?
Well, I just have a very, very, very general question.
Please.
Or maybe it's a statement.
Okay.
I mean, it seems to me that humanity as a whole just hasn't been very wise when we got a hold of some really powerful technology. Like the most glaring example is nuclear technology. I mean, look at the state we're in now. We can be blown up at any moment. And it's not, doesn't seem to be getting any safer either, but probably the contrary.
Okay, can I summarize your statement and go on to other questions?
I want to say one more example.
Uh-huh.
You know,
I think,
said how the smallpox vaccine hadn't done any harm, which I that isn't true. But
no, there are people who have had bad reactions
no,
to it.
I'm not sure how accurate this is, but I heard that some vaccine, I don't know for what, was used a great deal in Africa, and the people who were actually getting the shots didn't understand the importance of sterilizing the needle between each person, and that possibly, in some cases, 10,000 people were vaccinated with the same.
Would it be okay if I took some questions? And because I'm not sure I could reproduce.
Can I just, I'm sorry, I just interrupt briefly. I forgot to announce the fact that Garcia Street Books are making Nina's book available at the back, and I think you're going to, after the question, is going to be signing the books.
But forgive me, I don't mean to be rude, but I would like to answer a few other questions. Would that be okay? Because you didn't really have a question, you have a statement to deliver. Okay, yes?
I'd like to know if companies that are developing and the breeders who are developing GM corn in particular are looking at or doing anything to guard against another Texas cytoplasm catastrophe.
Okay. The question is, are the companies that are breeding BT corn guarding against another Texas cytoplasm catastrophe? What he is referring to is that there was a blight that was, that corn plants, so back in the 70s and 60s, corn plants weren't, to make the hybrid corn, you have to have one strain of one genetic constitution and the other, the male parent and the female parent have to be separated. On an actual corn plant, there are male and females. So you have to, in order for it not to self-fertilize, you have to take away the tassel. People identified mutations that made the plants male sterile. So they were still female, fertile, but male sterile. That means that they didn't have to go through and cut off the tassels, which is called detasseling. Now, it turned out that that particular mutant, it's a mutation in the mitochondria, And it was particularly, made the plants particularly susceptible to a rust that was coming north. And that I think it was 1972 or so, 70, okay, maybe 70, I didn't get my year right, really knocked down the corn yield. What happened after that is that companies got really, this was in the days before Monsanto, bought so many companies, and now Pioneer is owned by DuPont. But the companies basically got very conservative. They stopped using those sterile strains, and they went back to mechanical detasseling in producing the hybrid corn. Yes?
You addressed in your lecture various points concerning crops that are beneficial to mankind, such as your sweet potatoes and rice and corn. You never mentioned any of the strange modifications that have been taking place, such as the addition of insect and fish genes into crops to change color or to extend storage life or to make them harder for transportation and things of that nature.
So the question is, why didn't I mention these? Okay, so the question is, I didn't mention modifications such as adding a fish gene or
insect genes, scorpion genes.
Scorpion genes. I don't know, I'm not familiar with that, but the insects, certain kinds of fish, and plants make certain proteins that change the way ice crystals form. They come to be called antifreeze proteins, but they really have a very interesting mechanism. Because basically what happens is that they bind to the developing ice crystal and prevent its growth. Now, they were originally isolated in fish that grow in very cold waters, like in the Antarctic. The seawater is below the freezing temperature of the fish. The fish's blood, as it were, okay? And so it's an extremely important survival mechanism. But it turns out that these kinds of proteins are they're not fishy or what have you. They are proteins that are made in insects, they're made in fish, they're made in plants. And you're referring to a project that never got off the ground because it didn't work. So a company, back in the probably early 80s, decided, I can't even remember the name of the company. I'm not sure it even exists anymore. Anyway, so they wanted to add these genes to tomato plants so that the tomatoes could be frozen. It didn't work. So but there are other modifications, like for example, the bacterial gene that actually nucleates ice crystal formation. And the idea there was to delete that gene. And that kind of fell on the horns of the early controversies. So people stopped developing them because there were demonstrators that, trashed the fields and all kinds of stuff like that. Let's take some questions from the back. How about way back there?
Are there people who are working on genetically modifying corn so that it could be more efficiently used to make ethanol?
Are there people working on genetically modifying corn so that it could be used more efficiently to make ethanol? I would have said yes, except they're actually modifying it to make a more efficient fuel than ethanol. Butanol turns out to be better. And yes, people are modifying not the corn plant so much as the process of converting sugar to a fuel that has a higher energy value.
Could you say who they are, where they are?
Well, I know for one that DuPont is doing that. Yes?
The other major food in North America that's being genetically modified are soybeans. It looks like maybe 80 to 90% of the crops now are Monsanto Roundup ready. Are you concerned at all that with just this monoculture, one plant, one seed, a new disease could come in and basically wipe out the crop or severely affect the whole crop.
The question is, with the genetic modification of soybeans, does that raise a concern about monoculture? Monoculture and a particular genetic modification are two separate issues. Monoculture is monoculture, and yes, it does indeed bear with it the kind of susceptibility that surfaced with that particular rust variety. That is a constant concern because organisms mutate. The rusts are particularly good at it. Monoculture is efficient, but it is vulnerable. The genetic modifications don't make it better or worse. Herbicide tolerance is a problem. Much of it is quite labile, and it can, in some cases, the spontaneous. So you can get herbicide resistance. What herbicides do is they affect pathways that plants have that people don't have, which is why they're not toxic to people. But the plant can fight back. It can mutate to be herbicide resistant. So that's a constant management problem. And again, as with the insect resistance, the best management tools are multiple crop rotation and multiple different herbicides. At the moment, there are a number of different ones, and how fast herbicide resistance emerges is the only question. And that's an ongoing
There's
problem.
a long-standing question there at the back.
Yes,
but also we should take one or two because you need to have time just to make some signings. So perhaps one or two.
Sure.
Okay. Earlier in your speech, you basically gave the analogy of vaccination of animals and humans to plants. And you compared the analogy essentially of animals and human vaccination to plants. Now, I can buy that. Okay, when we're talking about taking genes from, fish and putting them into tomatoes, okay, how does that compare as far as an analogy to, transgenic modification of humans? Because the same people that are arguing for lowering the gates of research and letting this all open are, oftentimes on public radio saying that, we need to genetically modify humans to deal with the consequences of pollution and an emerging population density problem. Are you a post-humanist? Do you believe that we need to genetically modify humans to deal with the consequences of our,
No. I think we need to deal with the pollution problem. Am I?
You don't want to give any more of an answer?
I'm delighted, but you were going to.
Okay, why don't you take one more question, then we should wrap up.
Here's somebody that's really eager back there.
You've been very forthright that you are a proponent of genetic modification of plants. I'm wondering how much of your research or projects that you've worked on, or your colleagues, has been funded by such of these companies like
Zero.
Monsanto?
Zero. Never got a dime from them, sorry.
None of your projects?
Not one.
None of your organizations that you're associated with?
Nope.
Thank you. Thank you very much for a great 3 lectures. Okay.